Hi, everyone. I'd like to thank the conference organizers and the session chairs for inviting Novartis to speak today about branaplam. Branaplam is an investigational oral Huntingtin-lowering therapy for Huntington's disease. Today, I'll review some of the preclinical and clinical data that we've generated for branaplam, and then describe our plans to begin a phase 2b clinical trial in people with Huntington's disease later this year. Huntingtin-lowering therapies are among the most promising approaches in development for slowing the progression of Huntington's disease. It's well established that mutant Huntingtin is the proximal cause of disease onset and progression, and the benefit of lowering mutant Huntingtin has been demonstrated in numerous animal models. Because of this, many of the therapies currently in development are focused on lowering mutant Huntingtin protein. So what separates branaplam from the Huntingtin-lowering therapies already evaluated in people with Huntington's disease is that it is given orally. Thus far, all of the antisense oligos tested are administered via repeated intrathecal injections, and the current gene therapies are administered by one-time intracranial injection. Branaplam will be the first oral Huntingtin-lowering compound to be evaluated in HD patients. So how does branaplam work? Well, the full-length Huntingtin mRNA contains 67 exons that are normally all spliced together during transcription. During this process, the intervening introns get spliced out. Branaplam disrupts this process by promoting the inclusion of a pseudoexon during transcription between exons 49 and 50. Because this pseudoexon introduces premature stop codons, the Huntingtin mRNA degrades before it gets a chance to be translated into protein. The result of this is a decreased production of both normal and mutant Huntingtin protein. To demonstrate this effect in vitro, we assessed the impact of branaplam on the neuroblastoma cell line SHSY5Y. Starting on the left panel, branaplam promotes a dose-dependent increase in the expression of this pseudoexon in Huntingtin mRNA. In the middle panel, you can see that branaplam also dose-dependently decreases the expression of Huntingtin mRNA. And finally, on the right, branaplam dose-dependently decreases the expression of Huntingtin protein as well. So what happens in animals? Well, we dosed back HD mice orally for 1 week with branaplam, and then measured the inclusion of the pseudoexon in key brain regions. While vehicle-treated animals do not have any detectable Huntingtin pseudoexon, branaplam induces a dose-dependent increase in this pseudoexon in the striatum, cortex, and thalamus. Next, we dosed back HD mice for 3 weeks and measured levels of mutant Huntingtin protein in key brain regions either after 24 hours or 72 hours after the last dose. In both the striatum and cortex, we measured a dose-dependent decrease in mutant Huntingtin protein, and we found that the amount of lowering was higher 72 hours after the last dose compared to 24 hours. In this particular study, after 3 weeks of dosing, we found a maximal lowering of mutant Huntingtin protein of more than 45%. As we have previously presented, branaplam was originally developed for use in infants with type 1 spinal muscular atrophy, or SMA. SMA is caused by a mutation in the SMN1 gene, which leads to reduced levels of SMN protein. Branaplam restores appropriate splicing of a second gene, SMN2, which results in more full-length SMN2 protein. We have studied branaplam in infants and young children with SMA for more than 6 years. Branaplam has demonstrated good safety and tolerability in these kids and improves motor function and attainment of motor milestones in comparison to the natural history of SMA. So in order to demonstrate target engagement for Huntingtin in humans, we received consent to test blood samples collected from our SMA patients to measure Huntingtin mRNA levels. The lower gray line on the right panel shows levels of Huntingtin mRNA containing the pseudoexon. As expected, Huntingtin mRNA containing this exon was not detected prior to treatment. After branaplam treatment, mRNA with this exon was detected and remained elevated throughout the sampling period. The blue and red lines show Huntingtin mRNA detected using primer pairs upstream or downstream of the pseudoexon. Expression of Huntingtin mRNA decreased 25 to 50% during branaplam treatment. Taken together, the decrease in Huntingtin mRNA and the increase in the pseudoexon provides a proof of mechanism and target engagement for branaplam as a Huntingtin lowering agent in humans. In order to inform our dose and dose regimen selection for our upcoming trial in adults with HD, we did a phase 1 randomized, double-blind, placebo-controlled, sequential single ascending dose study in healthy volunteers. We found the elimination half-life of branaplam to be between 82 and 120 hours, which supports weekly oral dosing in adults with HD. We also demonstrated dose-dependent target engagement, shown by inclusion of the Huntingtin mRNA pseudoexon in blood. And lastly, we found that both Huntingtin mRNA and Huntingtin protein levels were reduced in blood after a single oral dose of branaplam. We have now finalized the protocol for our first trial of branaplam in people with Huntington's disease. The title of this phase 2b study is a randomized, double-blind, placebo-controlled dose range finding study with open label extension to evaluate the safety, tolerability, pharmacokinetics, and pharmacodynamics of LMI-070 branaplam when administered as weekly oral doses in participants with early manifest Huntington's disease. The goal of this study is to identify a safe and well-tolerated dose of branaplam that lowers mutant huntingtin sufficiently in CSF to expect a clinical benefit. This selected dose will then be evaluated in a larger phase 3 study to show a slowing of disease progression. The overall design of the study is shown here. So we will be recruiting male and female patients between the ages of 25 and 75 who are clinically diagnosed with early HD. Also, they have to have a TFC score greater than 8. Branaplam or placebo are given once a week as an oral liquid. The study includes 2 parts. Part 1 is a dose finding period, and part 2 is an open label extension. In part 1, we will use a staggered dose design in which we start with the lower doses and then introduce higher doses to subsequent patients. 80% of patients will receive branaplam and 20% will receive placebo. All patients will stay on their assigned dose and treatment until all patients complete the 16 weeks period. At that point, we'll have an interim analysis to select the dose for the open label extension, and all patients will enter part 2 and be reassigned to the selected dose. This slide illustrates our staggered dose design. We will have 3 cohorts, each with 25 participants, and randomized active to placebo in a 4-to-1 ratio. We start with cohort 1, our low dose, and then open up cohort 2, our mid dose, after the independent data monitoring committee reviews the data at our first cohort gating assessment. Once we have sufficient exposure to the mid dose, we have our second cohort gating assessment, where it will be determined if cohort 3 will include a higher dose, a dose in between the first 2 doses, or a dose lower than our first dose. The cohort gating decisions will be based on safety, not efficacy, but will include excess lowering of Huntingtin protein in blood as a potential safety measure. As mentioned, we will perform an interim analysis after all patients have completed the 16-week dose range finding portion, and all participants will stay on their assigned dose in a blinded extension until we select a dose for the open label extension. The 2 primary objectives of this phase 2b study are to assess the dose-response relationship of branaplam over 16 weeks on the change from baseline in mutant huntingtin protein in CSF, and to evaluate the safety and tolerability of branaplam when administered weekly for 16 weeks or longer in participants with Huntington's disease. Our main secondary objective is to assess the pharmacokinetics of branaplam on clinical, imaging, and biomarker endpoints relevant to HD. The key clinical assessments are shown on the top of this slide. They include standard HD clinical measures, such as cognition and apathy, as well as safety assessments, brain MRI, and lumbar punctures in order to collect CSF. The CSF biomarkers include mutant and total huntingtin protein, NFL, and a variety of exploratory biomarkers. From blood, we will measure mutant and total huntingtin protein, NFL, huntingtin mRNA, and exploratory biomarkers. We will also measure changes in brain atrophy by examining volumetric MRI of whole brain, as well as regions of interest, including caudate and the ventricles. So we are currently expecting to have our first patient first visit by the end of this year. And you can see here the countries that we plan to have participating in this first study. So we believe there are many advantages of a huntingtin lowering therapy that can be given orally compared to other administration routes. Most obvious is the ease of administration. A weekly oral medication is less invasive than intrathecal or intracranial approaches and can be administered at home without the need to travel to a specialized clinic. There's also the flexibility to start and stop as needed. Any side effects should be quickly reversible, in contrast to both the longer lasting ASOs and the irreversible gene therapy methods. While oral administration, we expect a uniform huntingtin lowering throughout the brain, and there may be potential benefits from lowering mutant huntingtin in the periphery. And lastly, in the future, it may be possible to monitor huntingtin lowering in blood only rather than CSF, which would be less invasive. So in summary, branaplam is an orally available small molecule that lowers normal and mutant huntingtin protein and mRNA. It works by promoting the inclusion of a pseudoexon in huntingtin mRNA. Following oral dosing, branaplam gets to key brain regions to lower mutant huntingtin in HD models. Chronic branaplam lowers plasma huntingtin mRNA levels in SMA children, and a single dose of branaplam lowers huntingtin mRNA and protein in healthy adults. In addition, the PK of branaplam supports weekly oral dosing in adults. Branaplam has been studied in babies and young children with type 1 SMA for more than 6 years and is well tolerated and efficacious. And finally, we're very excited to begin a phase 2b study in people with Huntington's disease at the end of this year. So on behalf of the entire Novartis branaplam team, I thank you very much for your attention. We are also very appreciative of the volunteers from our phase 1 and SMA trials. I'd also like to thank the investigators preparing for the phase 2b trial, our collaborators at CHOP, and our very distinguished HD clinical development advisors. Lastly, a big thank you to the HD COPE for providing insights into making our trial as patient-centric as possible. I'd also like to thank the EHDN Executive Committee for endorsing our study protocol, and to the Enroll-HD Clinical Trial Committee for accepting our protocol for the purposes of using Enroll-HD resources. Thank you all.